Welcome back to the HBL seminar. More like welcome back for me. I wasn't here last week, so hopefully it was a good seminar while I was away. But thank you for joining us again today. Today we have the pleasure of hearing from Taiki Ino, who is a PhD student here in kinesiology at the University of Calgary under the supervision of Dr. Walter Herzog. Before coming to Calgary, Taiki did an undergraduate degree in physical education, which he completed in 2015 at the Tokai University in Japan. After which he did a master's degree also in physical education from 2015 to 2017 from the same university in Japan. And his master's thesis was titled "The Effect of Maximum Voluntary Isometric Contraction Immediately Before Drop Jump on Drop Performance." Since 2024, he has been working here in the Human Performance Lab on his PhD under the supervision of Dr. Herzog, and his research involves some myofibril experiments and individual sarcomere behavior. Outside the lab, Taiki enjoys quite a few activities. This includes track and field, snowboarding in the winter, camping in the summer, and he also started learning long track speed skating for the first time last year. Back in Japan, he also enjoys collecting ceramics and stones such as crystals, so you should ask him to show you his collection. Today, he will be sharing his research on sarcomere length and force behavior in skeletal muscle myofibrils. Thank you, Taiki.
Thank you for introduction. So,
You need a mic?
yeah.
No.
No?
Okay.
Okay.
Can you hear me? Okay, yeah. So let me jump into the background. Little more mic. Oh, okay. So, or okay. So can you hear me? Or okay, yeah, thank you. So let me jump into background. For all people here, I think the skeletal muscle is very familiar. So just let me mention about the muscle hierarchy. So looking at the muscle, the whole muscle is bundle of fascicle. Then fascicle is bundle of muscle fiber. The muscle fiber is bundle of myofibril. So this is where I'm working on. So muscle fiber is very thin filament. So it's just around 1 micrometer. But micrometer is really hard to imagine. Just we can think it's really tiny. So let's compare with our hair. So looking at your hair or your friend's hair, it's just around 0.1 millimeter. So it's mean 100 micrometer. So myofibril is 100 times thinner than our hair. So looking at myofibril, there are sarcomeres aligned in series, and we can distinguish one sarcomere to the disc. And in one sarcomere, there are important proteins for force production. One is elastic filament titin, and this protein titin can produce passive force. So it means when sarcomeres stretch passively, they produce more and more force, like elastic band. And there are other important proteins, the thin filament coactin and the thick filament myosin. So on the myosin, there are many myosin heads which can attach to actin, so they can form the cross bridges. So by cross bridge cycling, they can produce active force. So between actin and the myosin filament overlap zone is important for active force. For active force, there is a relationship between sarcomere length and the active force. This is called force length relationship. And x-axis in this figure is sarcomere length, and the y-axis is normalized active force. So depends on sarcomere length. Amount of active force can be different. So first, looking at sarcomere length around 4. And this is schematic image of the sarcomere at this length. So at sarcomere length 4, there is no overlap zone between actin and myosin, so they cannot produce active force. But when we're looking at shorter sarcomere lengths, the overlap zone between actin and myosin increase, so active force also increase. Then this area called, this length called descending limb. And then this length cross bridge interact the most. So sarcomere produce active force the most. This length called plateau. From this length to shorter length, overlap duplicate. So the other side of actin interrupt cross bridge cycling, so sarcomere cannot produce active force like maximum. And this length is just around myosin length, so myosin can be slightly compressed, but sarcomere cannot produce active force as much as longer sarcomere length. So this is the ascending limb. So active force depends on sarcomere length and the actin myosin overlap zone. However, there is some phenomenon which cannot explain with force length relationship in descending limb with asymmetric contraction. So it's called sarcomere length non-uniformity. According to force length relationship, at the sarcomere length around 2.7, myofibril can produce around 70% of normalized force. But in the reality, when sarcomere in myofibril are activated in descending limb, sarcomere lengths tend to be not equal. Based on force length relationship, longer sarcomere produce less active force than other shorter sarcomeres. But sarcomere in myofibril are aligned in series, so they share the same force. This non-uniform sarcomere length distribution can be seen in both isometric contraction and the isometric contraction after active stretch. So people suggest that passive force from tightening contribute to compensate for the force under sarcomere length non-uniformity, which is called popping sarcomere hypothesis. Under sarcomere length non-uniformity, the longest sarcomere can produce active force the least. So we can say the longest sarcomere is the weak sarcomere. When the myofibril is stretched in active phase, the weak sarcomere stretch over the force length relationship to compensate for the total force by passive force. So the distribution of sarcomere length non-uniformity becomes bigger. This hypothesis can be cause of sarcomere damage from overstretch. However, it has not seen in experiments, and the sarcomere length non-uniformity cannot be explained with this mechanism. The origin of sarcomere length non-uniformity remains unknown. Now there is a question: why longer sarcomere keep the distribution and produce the same force with other shorter sarcomeres? There is an additional force on longer sarcomere to keep sarcomere length and force and prevent popping. But where does the additional force come from? In my hypothesis, in my research, we hypothesized that tightening contributes to give additional force, but in the sarcomere length within force length relationship, passive force is still tiny to contribute additional force to compensate. So for filling this gap, there is a hypothesis called tightening-acting interaction. So it can explain with this right side figure. The figure A shows the filament in passive state. So there is no tightening-acting interaction. When sarcomere is stretched, it produces passive force with full length of tightening. But looking at figure B, once sarcomere is activated, some part of tightening bind to actin. So free tightening parts become shorter, so it can produce higher passive force or produce passive force in shorter sarcomere lengths. So we hypothesize that this is the one of reason why longer sarcomere under isometric contraction do not elongate further. Go back to the graph. So if tightening-acting interaction happens in isometric contraction, higher passive force will be produced in shorter sarcomere lengths. It may contribute as additional force for longer sarcomere to keep the balance of force between shorter and longer sarcomere and prevent popping. So purpose of this research is investigate the mechanism responsible for preventing sarcomere length overstretching in isometric contraction. Our hypothesis is tightening-acting interaction likely protects sarcomeres from overstretching in isometric contraction. To test this hypothesis, we deactivate one sarcomere during isometric contraction. But how deactivating one sarcomere helps to test this hypothesis? Let me explain with the experiment image. So this is schematic image of sarcomeres in myofibril with experiment setup. Red line is actin, and the orange oval is myosin, and the black line is chitin, and green vertical line is dead disk. So under there is micropipette with relaxation solution to deactivate only one sarcomere. And this is large bore pipette to activate all sarcomeres in myofibril. So in this protocol, we first activate all sarcomere in myofibril, so they produce active force. And also we expect there is tightening-acting interaction happens. And then deactivate one sarcomere. The sarcomere do not produce active force, so the sarcomere needs to compensate for the force with passive force. But tightening-acting interaction may produce higher passive force in shorter sarcomere lengths, so the sarcomere will not pop. This experiment can show if tightening-acting interaction contribute to the sarcomere protection. But what happens if our hypothesis is wrong and the tightening-acting interaction does not happen in isometric contraction? So even though we activate sarcomeres, there is no tightening-acting interaction. So when one sarcomere deactivated, it will elongate further for the compensate for the force. So this is a test for our hypothesis. For this experiment, we use myofibril, so we need muscle sample. So we take sample from New Zealand white rabbit. We take piece of sample from thorax and then tie it up on the wooden stick. then skin them 14 days. Then it will be ready for experiment. So the day of experiment, we homogenize sample. Then we take sample single myofibril and set up for the experiment. For this experiment, we use special protocol. It's called jacuzzi protocol. Name is very gorgeous and relaxing. Thank you. The reason why this protocol is called jacuzzi is that we give two types of jet to myofibril. So myofibril is taking jacuzzi bath. But this jacuzzi is very small. The inside diameter of this bath is just around 1.7 centimeter. So this bath is only for myofibril. We need to set up using those four listed equipment in a small bus. Sorry. So first, this is cantilever, which can measure the force with displacement of one side of the lever. This is needle for picking up myofibril and control sarcomere length. And between needle and the cantilever, myofibril is folded. There are two pipettes. One is micropipette, so it's filled with relaxation solution, so we can target one single sarcomere to deactivate. So micropipette tip is just around 1 micrometer, so it's very tiny. Then there is other large ball pipette to activate all sarcomeres in myofibril. And looking at the scale bar in right picture, it shows 50 micrometer. So roughly width of this picture is around 100 micrometer. So I need to set up those all four equipment within the width of the hair, thickness of hair. So it's very tiny work. And for observing and setting up this tiny experiment, we use optical microscope with manipulators. The left picture is whole system. There are four manipulators on microscope to control the location of cantilever, needle, and the two pipettes. And on the center of the manipulator, there is a white board to set the experiment bus. In the protocol, I need a micro-level control for all equipment. So there are also finer manipulators to realize it. So those are set up for my experiments. So from now, let me talk about my experiment progress so far. So this is the first experiment I've successfully done after many trials. So it's recorded around last year, April or May. So as we can see here, this is cantilever, and we can see the displacement of one side of liver. Then we can measure how much force the myofibril produces. Then myofibril is attached with the needle. And on the center, this is micropipette filled with the relaxation solution. So I deactivate one sarcomere with moving up and down micropipette. And then we cannot see from here, there is large ball pipette on the behind to activate all sarcomere. By the way, then I tried many experiments, but I figured out I cannot run the experiment constantly because there are two main problems. So one is the activation of a single sarcomere. And the other one is individual sarcomere length measurement. So first, looking at the activation of a single sarcomere. So what's the problem with the deactivation? So while I tried many experiments, I found difficulty to deactivate only one sarcomere. Sometimes I deactivate few sarcomeres, and then other time I deactivate only neighbor sarcomere instead of target sarcomere. So for figuring out what makes deactivation light and weird, I tried two things: the micro-injector femtojet and the visualized solution flow with methylene blue. So first I'll start with femtojet. Sorry. Yep, after I did many trials, I figured out micro-injector, the femtojet works well for the protocol. It can push and stop the solution, as we can see in this video. Also, it can control tiny amount of solution delivery. Actually, I don't know how much solution it pushes, but as we can see in the picture of micropipette, it filled with 5 microliter of solution, and it will work almost forever. However, even though we develop tiny micropipette and the technique of micro solution delivery, I still deactivate few sarcomeres. So I need to make sure how the solution goes in this experiment setup. So now we figure out the solution delivery with femtojets. So let's looking at methylene blue test with solution flow. So I visualize micropipette solution with methylene blue for checking solution flow. So we can see it in as dark solution in this video. First, I only push solution from micropipette. So as we can see here, so it's middle spreaded, so it may affect 2 to 3 sarcomeres. Then I push other solution from behind with large bore pipette, and it gets thinner enough to affect only one sarcomere. After that, while keeping solution flow from large bore pipette, I push more solution from micropipette. Then the solution flow gets thicker. But I push more solution from behind, and it can be thinner too. But strong solution flow push and bend the myofibril. So the balance between the strength of solution flow from micropipette and large bore pipette is important for affect only one sarcomere in the myofibril in this experiment. So now we measure how affect single sarcomere. Then we have one more problem, individual sarcomere length measurement. So what is the problem with the measurement? So in myofibril mechanical test, this phase contrast image is commonly used. It emphasizes the visibility of transparent specimens, as we can see in this picture. This image shows passive states of myofibril. We can see the striation pattern of myofibril and zip disks. So it seems like no problem to measure individual sarcomere lengths, but there is a problem of zip disk visibility in active states. So once myofibril is activated, we cannot see zip disk. So we are not able to measure accurate individual sarcomere lengths. Like here, we are not able to see this. For solving this problem, we use alpha-actinin fluorescence. Oh, sorry. Why though? Yeah. So bottom video is phase contrast image, and the top video show alpha-actinin fluorescence. Alpha-actinin is protein which is component of Z disc. So we are able to label only Z disc. This technique allows us to track Z disc location and the individual sarcomere length while either active and passive states. So now I complete both troubleshooting for this experiment. However, it is still tiny scale and delicate. So I have successfully done just 4 experiments so far. So it means the sample size of targets that deactivated sarcomere is 4. Neighbors sarcomere, the one sarcomere each right and the left side of target sarcomere are 8, and the other sarcomere are 48. There are still not enough data, but I will show you the trend of the experiment. So the top figures show the sarcomere length change, and the bottom shows stress on the myofibril. And both figures share the same timeline. And the timeline at 0, the all sarcomere activated. Then right away, the target sarcomere in this experiment, the number 4 sarcomere, so blue line, it's elongates around 0.6 micrometer, while other sarcomere keep the sarcomere length well. Then once it's reactivate, it's able to shorten again. And looking at stress, at the same time, the stress dropped from 310 to 280 nanonewton per micrometer square. So it's around just 10% reduction. Then once the target sarcomere reactivate, the myofibril able to reproduce almost same amount of force. Then this right figure show the absolute sarcomere length with the initial sarcomere length. The initial sarcomere length means before activation, so it's passive state. So looking at passive state, all sarcomeres stay around 3.3 micrometer, but once it's activate, they show sarcomere non-uniformity. And we deactivate one sarcomere, so its target sarcomere is this blue line. So it's elongated, but it did not pop. It did not stretch over the force length relationship length. So it's able to come back to similar lengths. Then this is all for experiment data. So top figure show the sarcomere length, and the bottom show the stress. Then this two figure also share the same timeline, but this time there is 3 time points: full activation, and deactivation, and the reactivation. So first looking at neighbor sarcomere and the other sarcomeres, the orange and the green circles. All the time points, they keep same sarcomere length, so we can say it did not deactivated. But looking at target sarcomere, the blue circle, once it's deactivated, they elongate around 0.6 micrometer. But after when it's reactivated, it's able to shorten again. And looking at stress, when target sarcomere deactivate, they reduce the stress in the myofibril, but when target sarcomere reactivate, the myofibril are able to reproduce similar amount of force. So let's looking at only target sarcomere behavior. So this figure show only target sarcomere behavior. The x-axis show sarcomere length, and the y-axis show the stress. In this figure, both green and the blue marker of the same individual sarcomere in passive state. But blue is pure passive. It means before activation. The green is passive state after activation. So both green and blue force come from passive force. In compared to blue markers, green produce higher passive force around the same sarcomere length or shorter. If the activated sarcomere need to compensate for the force without additional force from Taiki-actin interaction, even sarcomere length 4.5 cannot compensate the force produced with deactivated sarcomere. In fact, the deactivated sarcomere did not elongate over the force length relationship. Based on this result, sarcomere length non-uniformity within myofibril could be explained by Taiki-actin interaction, which allows sarcomere to produce the same total force with different sarcomere lengths. But we still do not have enough data to conclude. So for the future plan, we will do more experiments for more data. And then once we did enough experiments, we will do next experiment, which induce popping in isometric contraction. So let me explain future experiments. So this is the same schematic image with before. So in our hypothesis, once sarcomere are activated, Taiki-actin interaction happens. So if we keep relaxed one sarcomere while other sarcomere activated, we hypothesize that the relaxed sarcomere elongate further to compensate for that force without Taiki-actin interaction. Then we can prove that activation is a key component for the Taiki-actin interaction to keep balance of active force with different sarcomere lengths and prevent sarcomere from popping. Protect sarcomere from popping. So as conclusion today, we have provided pilot evidence that Taiki-actin interaction likely protects sarcomeres from overstretching in isometric contraction. So this is what I am doing and what I will do here in HPL. So this is the reference for today's presentation. And thank you for Dr. Walter Herzog and all member in Herzog member. And thanks to those all organizations to help my experiment and research. Thank you.
I have two questions. The first one is, why do you care about deactivating a single sarcomere? I mean, if you can deactivate a few and still measure an overall length across several, that almost as good as deactivating a single one?
Yeah, thank you for that question. So we expect if we deactivate several sarcomere, so we usually measure force, the myofibril force with displacement of cantilever. So if we deactivate, they will not, let's say, if I deactivate just one single sarcomere, even keep deactivation, they still keep some stress on myofibril. But if I deactivate few sarcomeres, probably they elongate a lot, so they will not keep the tension in the myofibril. So probably we expect if myofibril lose tension, they cannot keep the tightening actin interaction. So we focusing on to deactivate one single sarcomere.
Okay, and my second question is, so you showed your titin actin interaction graph. And in that graph, you kept the sarcomere at the same length right there. So there's a same length, high force and low force, right?
Same,
So you have the difference there. And
yeah,
that's taking a hypothetical sarcomere, keeping its length constant, and then imagining what the force could be, and therefore there's something compensating.
yes.
And my question is, since you showed that the sarcomere length increases by 0.6 micron or something like that, shouldn't that picture be sort of redrawn to take that into account?
Oh, sorry,
Or maybe the question is the 0.6 consistent with this picture? So I guess what I can't tell here is where was the starting length compared to the increased length? Was that 0.6?
so mean like the force curve starts from here or sorry.
Well, so
Yeah.
it looked like you had a range of changes in the sarcomere lengths, and then the target sarcomere changed by a length of 0.6. And I'm just wondering if what you're depicting here is consistent with that 0.6.
So it actually they show different lengths because also they also produce different force. So when we deactivate it, some sarcomeres show less elongation, but sarcomeres show more elongation. So it's not constant on distance, 0.6. Just it was average.
Okay, and then maybe still the question is, for your depiction of titin actin interaction, is that picture consistent with your data now that you have the data?
So here,
So what you showed here
yeah,
is kind of it's a conceptual idea of what you think will happen.
yes,
That's before you had data.
yes.
Now you have data. And the question is, does your picture change in order to agree with the data, or did you have it exactly right?
I see. So we still don't know how how much amount of the passive force from tightening actin interaction contribute to the different sarcomere lengths. So we cannot still say exactly it's 100% compensate the force, but still, so our results show it produces higher passive force. So it seems like it's the tightening actin interaction happen and then help to compensate the force.
Well, I wonder, since you have 4 experiments, you actually have 4 data points, right?
Yes.
And so you could imagine 4 yellow dots instead of the one that you're pointing to. And then if those 4, let's pretend they're accurate. Then those 4 should show you a trend for the amount of sarcomere length change and the amount of, supposed force that's being compensated. And so shouldn't that also contribute to your, I don't know, evaluation of titan actin interaction?
I see. Yeah.
So question.
Also,
So I think the question that Art is asking is, can you use those 4 data points that you have now to then estimate whether this depiction is correct or not? Is that part of your future plan to do that?
yeah, probably, yeah, in the future, I want to figure out how it works, but probably it depends on sarcomere length. I expect like in shorter, if it's activate in shorter length, they produce the titan actin interaction in different place. So probably it could be different passive force line. So I'm still thinking how to make with like a different amount of titan actin interaction passive force.
Thank you.
Brent, you had a question?
Can you bring up that slide again that shows the different lengths of the sarcomeres? No, it's the one that has the experimental data.
I think the one.
You
with the
activate,
target.
deactivate, you've got the.
Yeah.
No, the other one. There. There, thank you. So you activate, you deactivate at time point 0, right? That's when you relax at time point.
Right.
What's responsible for the non-uniformity across all the sarcomeres before that? So you activate and some lengthen and some decrease. And they're all decreasing and lengthening at different amounts.
Right, so still we don't know what happened to the sarcomere length non-uniformity. So, but I expect, so those shortened sarcomere produce active force, but those lengthened sarcomere produce active force with the passive force. So they compensate some part of active force with the titan actin interaction.
So that has nothing to do with sort of the amount of proteins that are inside each sarcomere or the amount of activation solution that's being delivered to each sarcomere.
Could be there is like each sarcomere has different the titan or some filament difference, but we cannot observe by this mechanical test.
And then what's happening to the red one on the very top that's going past this line that says the pocket?
Yeah, I think, yeah, probably they produce quite high passive force in active states. So it means they are producing higher passive force than pure passive force at sarcomere length 4. So it means they are probably producing titan actin interaction once it's activated. So they will elongate. So they produce the higher passive force. But here there is no activation force can produce. So only they produce the passive force with titan actin interaction.
So how do you come to the conclusion that that top line has a titan actin interaction? Why can't it? And I think you talked about it a little bit before in the introduction, but I've already forgotten. So how do you know that that's just not pure passive force production?
I see. So for example, here at the time 0, they produce around 280. So no, it's 110 for activated. But when we compare with the pure passive line, so around 280 mean the sarcomere need to elongate over 4.5 to produce the amount of force with pure passive force. So probably the sarcomere produced higher passive force, but it's over the force length relationship. So they produce passive force, but it's not pure passive force.
That makes a lot of sense. Two more really quick questions. Back to the other slide.
This one?
Yeah, yep. No, the other one. I've lost it. The one, yeah, there, okay. So if you deactivated one of those sarcomeres and it just happened to go above the ... popping.
Yeah.
Would you have a different interpretation of your data? Cause you looked at the blue line, which is the one you relaxed,
Right.
and you said, it went up, but it didn't pop.
Mhm.
I think you said that.
Yeah.
But if it was a different sarcomere that you activated, and then you deactivated, it could have gone above the popping line.
That's right. So probably if we deactivate those sarcomeres, probably they will be around the popping line. But also they are already, I think they are already producing force with the titan actin interaction. So even we deactivate, so it's just reduction of maybe few percent of active force. So they will not elongate as much as those supplements.
Venus, you say on this slide, the sarcomere, the blue one,
Yes.
it seems like in the first figure, like the first on the top, our left, yes. So it seems like it was going to fall, like that, the change of light, it seems it has, different regions. So maybe initially it was starting to fall, and maybe at that point, or that the titan was stretched at that point so that it starts to resist the popping. You think that? So what's your interpretation of having a
This-
very steep first change in length and then less?
That's a good question. I also curious about this one because when I check other experiments, they show just very flat. After they show steep elongation, they keep flat length. But this one show the little moderate elongation. So actually, I have no idea what difference between the steep and the moderate deactivation.
Any questions for Taiki? Brent, yeah, Beto, and then I'll come back to Brent.
So acting titan interaction seems to be important.
Seems to be important. Yes, I believe. Why? So, so I think, so it's, so for example, like so this case, like this is asymmetric contraction. So then we expect it's like a protect the titan actin interaction, protect from overstretch. So it's mean like if it's stretched and then like yeah, when it's stretched like a force according to force length relationship, they produce less and less active force. So if we think about our body, when it's stretched, then if we lose more and more power, and then if I get more tension to go the other way, it could be the injury for our body. So, so, uh, so yeah, preventing the overstretch with those passive elements is important for our body for protecting from injury.
You know, I was sitting on the sidelines. I'm not a muscle person, but I've about 50 years now, I watch development. And it's basically a development from a relatively simple model to a relatively complicated model. And now you have to bend backwards to explain with all these different things to explain that the curve at the end goes up and not down. I think that is exaggerated, or do you see that as a simple solution of the situation?
Simple solution for the situation. So,
But in principle, you have at the end, the curve, instead of going down, curve comes up. You want to explain that the point should be on that.
so it's clear.
Yeah, that, you know, you, at the bottom right.
Yeah.
It's, they are not there, this point, so they come up again. So you need something in the end, which you manipulate the whole thing then to get that.
I think what the question is like, do you think that the work you're doing is a model or whatever the model has come to now is a simple or a complicated solution to answer what's going on at that end?
I think this is very straightforward hypothesis.
Perfect, Brent.
I have a very naive question, and I hope Venus doesn't laugh at me. I just wanna know, are we sure, are we positive that at this length scale, there are no protein interactions that cross Z disks? We're sure that everything happens in between these two Z disks. There's nothing at this length scale that can cross from one sarcomere to another.
So your question means there is some like a connection between the sarcomeres? Like,
Yeah, my question is like, are we sure that there isn't one? Like, there's not some protein that goes, just bounces, that cross hundreds of at the myofibril level. So you say there's a passive element, titan, that's keeping it from being stretched.
Yeah.
But what if there's another element that's 10 sarcomeres away that's keeping it from being stretched?
I'm not sure if I answer your question, but like in the future experiment, I try to do this one. Like I try to deactivate one sarcomere, then probably it will show the popping. So it means like that one target sarcomere is deactivated and then they behave very individually. But if this experiment show like a less elongation, probably there is some relation between each sarcomeres. So we will see it. Is it?
I mean, is that a dumb question?
No, it's not, but we do not know. So far, we believe that it's titan that is responsible for passive force within this sarcomere length. There are intermediate filaments, but in myofibrils, we believe they're not there anymore. and they do not work at sarcomere lengths below 4, let's say, they are connecting one Z line to the next to the next. And so, until we have discovered other proteins, we do not know. That's it, no, that's it. We have an online question?
Yeah, go ahead, Walter. Walter,
Okay, this is me. Can you hear me?
we can hear you clearly.
Okay, good. Yeah, I just wanted to make a couple of comments to some of the questions that were made. And, initially, why do we deactivate one sarcomere and why not many? The very simple answer is that the initial idea was that we would deactivate one sarcomere and then 2 and then 3 and then 4 and see what the dynamics of the sarcomeres would be if we did that, and also what the force loss would be, because Azim and I wrote a theoretical paper on that and have a very distinct prediction about it, but it seems to be at the moment not possible to deactivate one sarcomere with the setup that we have and then keep it deactivated while deactivating a second and then a third and then a fourth, because when we go away with the deactivating solution and the little pipette, then immediately the sarcomere will be activated again. So in fact, the idea was that we wanted to do that, but technically we are working on being able to do that, but at the moment with Taiki setup, we cannot. The other comment that I wanted to make is that, the question was in the conceptual figure that Taiki has, should the sarcomere become longer? And yes, it elongates a little bit. And, but the key to remember here is, and that conceptual figure is actually a bad figure because it has a very steep passive force at some point compared to the decrease of active force on the descending limb of the force length relationship. Whereas in reality, the increase in passive force is much slower with a given sarcomere elongation than the decrease in active force. So if you increase by 0.2 microns or by 0.4 microns or by 0.6 microns, if you take purely the passive force of the sarcomere, it cannot compensate for the loss in active force. There will always be a deficit. And so therefore, if it elongates, whatever amount it elongates, it will lose more active force than it gains passive force. And so something must be happening for not letting it slip and completely, what Taiki called to be popping. And then, Brent, with his question, hit it on the head. We just published, I don't know, Venus will remember 2 or 3 years ago about what is responsible for sarcomere length non-uniformity. And we did a whole series of experiments with Meng Li, a former MSc student of us. And the basic conclusion was that, yes, it highly likely has to do with different amounts of actin, myosin, and titin proteins in one sarcomere to the next. The interesting thing is that people have always assumed that in a myofibril, the protein content from one sarcomere to the next is constant, but I and I guess some other people don't really believe that. The interesting thing is nobody has ever checked that. And Chris Thiessen right now, who is an undergraduate student but will become a master's student, one of the many things that he tries to do is exactly measure that. How many proteins did we have in one sarcomere and the next one, the next one, the next one in a myofibril? I think we probably can explain those development of sarcomere length non-uniformities with the amount of protein content. And then one of the things that might not have come as clearly apparent from Taiki's talk as it might have been is the next set of experiments that we want to do. Just to recall what Taiki showed you, he showed you where a myofibril is fully activated, every sarcomere is activated, and then he deactivates one sarcomere and observe what's happening. The reverse experiment that we now want to do is that the whole myofibril is not activated. Nothing is activated. And we spray the deactivating solution at one sarcomere, and we keep it deactivated. And now we activate all the other ones. And the prediction is that now, since that sarcomere that we have deactivated never was activated, it did not form this connection between actin and myosin, between titin and actin, and therefore it's much, much softer and only has the passive titin properties, which are much, much softer than what we are seeing, and therefore this sarcomere will not be pulled 0.4 or 0.5 or 0.6 microns, but it will be pulled 2 or 3 microns way beyond overlap, and maybe, I think maybe the sarcomere, the myofibril might actually break. And so, and I think that didn't really become clear. So the idea really is we do these sets of experiments, and then we do those reverse experiments, and we compare the force loss and the sarcomere elongation between those two experiments. And the very simple idea is that in the reverse experiment, force loss and elongation of the deactivated sarcomere, the one that was never activated, will be infinitely bigger and look completely different, thereby indicating that you need to activate the sarcomere before it achieves that stability that we seem to be seeing. And then the question about, are there connections across Z disks? And yes, there are. I mean, proteins from one sarcomere meet the other one. And about 20 years ago, I wrote a paper where I said, well, a sarcomere is the functional contractile unit of a muscle, but I also argued it's not an independent contractile unit, like that the Z disc is a wall, and you pull on one side with one sarcomere, you pull on the other side with the other sarcomere, and there needs to be an equilibrium there if that wall doesn't move. I don't believe that, but also the experiments from Taiki showed that when you do something very dramatic from one sarcomere, where you have an active force of 300 units, and now you deactivate and goes instantaneously to 0, not much happens to the neighboring sarcomere. So even there appear to be connections across the Z disc. Functionally, what they do and how relevant they are, that I think that's a question that's not been answered. So those are just a couple of the comments that I wanted to add that maybe help a little bit understanding a bit more. And the other thing that I wanted to say to Taiki, I know you were very nervous, and I know you are worried about your English, but I thought that was a fabulous presentation. And I think the other thing that I wanted to say, in order to have that setup, is the micropipettes and the multiple activation deactivating solution. That's that's a technical marvel piece that Taiki developed there, and if nothing else, he should be congratulated for just making this actually happening and now being able to do those experiments. So that's all I wanted to say. Thanks again for everybody coming, and thank you, Taiki. I thought that was really great.
Thank you, Walter. And with that, I think we should thank Taiki.